I would like to welcome our next speaker. She is Azin Amin. If you want to start sharing, perfect. She is a third-year PhD student at the Florey Institute of Neuroscience and Mental Health in the University of Melbourne, Australia. And she now we are going really deeper into the neuron and especially into motor neurons. So please, Azin, the floor is yours. And if you have questions for the speakers, you can write them on the question and answer box, and you can also upvote other questions if you like them. So please.
All right, great. Thank you so much for the introduction, and thank you for giving me the opportunity to present my research. So basically, my project focuses on developing peptides to treat the neurodegenerative disease ALS, which is also known as MND. So I don't know whether anyone remembers, but this is from the ice bucket challenge, which was where people had to dump cold ice water over their head. And this challenge was designed to raise awareness for this disease ALS. So amyotrophic lateral sclerosis, or ALS, is similar to other neurodegenerative diseases in the sense that there is an accumulation of misfolded proteins into these clumps or protein aggregates. And these accumulate in the motor neurons of patients, which then contributes to the degeneration of motor neurons. As a result of this, all these signalings to muscles get cut off. And so muscles start to waste away. This muscle wasting spreads around the body, and once it reaches the respiratory muscles, it causes death in the patients. And this usually happens 2 to 4 years after the onset of symptoms. So what's more is that there is no cure for this disease, and the only 2 treatments available, which are riluzole and edaravone, they can only extend survival by a few months. So there is a great urgency in trying to find a treatment for this disease. Normally in cells, there is a process called autophagy, which keeps the cells healthy by degrading down these misfolded proteins, protein aggregates, dysfunctional organelles, and any other toxic agents. So autophagy has 4 important steps. In the first step, a pre-autophagosomal structure forms in the cytoplasm. Then a complex which has this Beclin-1 protein as its core component, this LC3 protein gets lipidated to LC3-2 and gets incorporated into the membrane. And then this complex fuses the membranes together to form the double membrane autophagosomes. This same Beclin complex is then involved with the fusion of autophagosomes with lysosomes and the formation of autolysosomes. It's then in these autolysosomes where the enzymes degrade down all of this cargo, and any generated macromolecules gets recycled back into the cell. So we are thinking, because in ALS there is an accumulation of these protein aggregates, we are thinking that this autophagy pathway might be downregulated or dysfunctional. So if we could find a way to upregulate this pathway, we might be able to degrade down these protein aggregates and prevent motor neuron degeneration in ALS. So to upregulate this pathway, we focused on this Beclin-1 protein. So Beclin-1 protein is the master regulator of autophagy because it's involved in both the early and late stages of autophagy. And a lot of proteins interact with these different domains to regulate autophagy. So for example, the 3 identified domains of Beclin-1 protein are BH3, CCD, and ECD. From previous research, we know that this ECD region of Beclin-1 on its own is sufficient to induce autophagy. So we wanted to make peptides derived from this ECD region. So this is the whole ECD region. So for the first peptide, we decided to focus only on this yellow bit, and we named it Beclin-1. And then we decided to extend it from the N-terminus and named it Beclin-2. For Beclin-3, we decided to include the whole antiparallel beta strand. And for Beclin-4, we excluded this random code. So it's just that beta, random beta code. Oh, sorry, the beta strand. So these peptides were designed and synthesized. But as many of you might know, so peptides are great for drug candidates because they're small and they're very selective towards their target. But a major limitation of peptides is that they cannot penetrate the cell membrane, and so they can't get into the cell. To help my peptides get into the cell, I used a fragment derived from this apolipoprotein E. Why did I choose ApoE to help my peptides get into the cell? Because ApoE, compared to the other cell-penetrating peptides, is safe because it's an endogenous protein, and on its own, it has shown protective effects in injury and neurodegenerative disease animal models. ApoE is also potent because not only can it interact with the anionic proteoglycans, but it also has its own receptors on the cell membrane. And because most of these receptors are in the brain capillary endothelial cells, they are more selective towards the brain. So that's what we want. So it makes it selective towards the brain. And also our lab has shown previously that ApoE can cross the blood-brain barrier, which is great. But another problem we encounter is that once the peptides get into the cell, they will likely get entrapped within endosomes and get degraded or recycled out of the cell. So to address this problem, we used the fragment derived from this hemagglutinin protein, and we named it HA2. So basically what will happen is that once the peptides get into the cells, they will get entrapped within endosomes. But then this HA2 peptide can disrupt this membrane and help our peptides escape into the cytoplasm and go and activate their target. So in the end, 13 different peptide analogs of Beclin-1 were designed and synthesized. Next, our question was whether they can upregulate or induce autophagy. To measure autophagy activity in cells, we use this reporter, which is the Emcee EGFB LC3 reporter. So I mentioned earlier that this LC3 protein gets incorporated and inserted into the membrane of autophagosomes. And because now in here it's tagged with EGFB and Emcee, autophagosomes will appear as yellow puncta. But as autophagosomes go on to fuse with lysosomes, the pH drops, and so EGFB gets quenched and only Emcee would be visible. So our autolysosomes will appear as red puncta. So by measuring the number of yellow puncta, we will know the level of autophagosomes, and by measuring the number of red puncta, we will know the number of autolysosomes. So if something is increasing autophagy, we should see more red puncta compared to yellow puncta. So we did that, and this is my result. So on the x-axis, we have the different peptides that were derived from this Beclin protein. And I have the two controls here. So rapamycin is my positive control. So this is the gold standard autophagy inducer and is currently in phase 2 clinical trials for treating ALS. And bafilomycin A1 is our negative control. So it inhibits autophagosome fusion with lysosome. So it prevents autolysosome formation. So as you can see, two of my peptides were able to increase autophagy significantly and even higher than our positive control rapamycin. And these are our controls. So basically, out of all the 13 different analogs that we synthesized, two of my peptides, HA2 ApoE Beclin 3 and HA2 ApoE Beclin 4, were able to induce autophagy. So our next question was whether they can reduce the protein aggregates that are in ALS. So ALS is mostly sporadic, but 10% is familial. And within those 10%, these are the genes that have been identified. These are some of the genes. But the most studied gene is the SOD1 gene, which encodes the superoxide dismutase protein, which is an enzyme in the body that converts harmful superoxide radicals into oxygen and hydrogen peroxide. But what happens in ALS is that these SOD1 proteins become misfolded and they aggregate. So I wanted to see whether my peptides can reduce these SOD1 aggregates. So I did Western blotting. And I looked at the insoluble fraction because that's where the misfolded proteins will go. So I looked at the wild type SOD1 and also the mutant SOD1. In this case, I'm looking at A4V. And as you can see, my peptides didn't have any effect on wild type SOD1, which is great, but they were able to significantly reduce the number of insoluble mutant SOD1 or the misfolded SOD1. To confirm these results, I also did confocal microscopy and I counted the number of cells containing these EGFB aggregates. And again, as you can see, my peptides were able to reduce the number of SOD1 aggregates. So these peptides can upregulate autophagy, they can reduce SOD1 aggregates. Our next question was whether that would translate to a beneficial effect to the health of the cells. So from previous studies, we know that mutant SOD1 activates a cascade of events, which leads to caspase-3 activation and eventually apoptosis. So I wanted to see whether the peptides can reduce caspase-3 activation. So in here, again, as expected, expression of this mutant SOD1 or A4V increased caspase-3 activation, but this was decreased back down after treatment with our peptide. And our controls here are HA2-ApoE scrambled Beclin 4, which is our negative control, and astarosporine, which is a drug that induces apoptosis, as our positive control. So in conclusion, HA2-ApoE Beclin 3 and HA2-ApoE Beclin 4 were able to increase autophagy in cells. And these were 2 to 10-fold more potent than the previously developed peptides. From this, we can conclude that the HA2-ApoE is required for the activity of peptide because Beclin 3 on its own wasn't able to induce autophagy, but it had to be conjugated to this HA2-ApoE, and the same with Beclin 4. And because Beclin 3 and Beclin 4 were most effective compared to Beclin 1 and Beclin 2, we can conclude that the inclusion of the antiparallel beta strands gives the highest activity. We also showed that the Beclin peptides can prevent mutant SOD1 aggregates, and as a result, they can reduce cleaved caspase-3 activation, which is mediated by mutant SOD1. And finally, I'd like to thank my supervisors and everyone at the MND lab, and thank you for listening.
Thank you very much, Azin, for your great talk. So, and for staying in the time. So thank you very much. Let's see if we have any question from the attendees. None right now, but I have one myself. So I'm kind of far away from your specific research area. What I wanted to ask is how, so as far as understood, those are in vitro studies. Are you expecting to translate this approach also in vivo to see the same but on an in vivo study?
Yes, so currently I'm actually working with LC3 mice, which works exactly as that reporter, but it's just in mice. And yeah, I'm injecting my peptides into mice to see an effect. And if I do see an effect of autophagy activity, if I see it in LC3 mice, then I'm gonna go into SOD1 mice and see whether that would have any beneficial effects in an ALS mouse model.
Okay, perfect. That's I think that's the direction where you want to go to approach in the end and kind of find a solution for this disease. Okay, so if we don't have any more questions, we are exactly ending at the time we are supposed to. So I would like to thank all the attendees.